Thanks, John, for
That's
giving
good.
me a chance to present here and for organizing this wonderful workshop. And hello, everyone. I'm Jiwoong Kwon, a postdoc in TechPath Lab at Johns Hopkins University. And today I'm going to talk about super resolution imaging approaches on the JCVI minimal cells. And this work is actively ongoing and has not reached well-organized result yet. Instead, I'd like to share the experimental details and the preliminary result, as well as some problems to be addressed. So yeah, I'd like to get any feedbacks from the minimal cell experts about this work. So yeah, please let me know if you have any questions, suggestions, or ideas. As we all know now, JCVI-syn 1.0 was reported as the first self-replicating synthesis cell, and subsequently, the SYN 3.0 was developed as the first synthetic minimal cell, having only 473 genes. But one abnormal behavior of SYN 3.0 was the heterogeneous morphology, which is not normally observed in the natural species. And James and co-workers found 7 more essential genes for the homogeneous morphology. And with these additional genes, SYN3A now becomes the minimal model for bacterial physiology. And these minimal cells uncovered the genes required for the minimal life activity, but still the functions of many of those genes remains unclear. And even for the well-studied genes such as FTSC or ZEPF, the intracellular distribution and the dynamics of those proteins inside the minimal cells are poorly understood. So in this project, we aim to visualize some proteins inside the SYN3A or B to provide structural details for a more comprehensive understanding of minimal cells. One major obstacle for the microscopic approach is the small size of the minimal cells. And SYN3A has about 500 nanometer of diameter, which is slightly larger than the diffraction limit. So we cannot well examine the structural details with the conventional diffraction limited approaches. So the main tool of this work is the single molecule localization microscopy, one of the super resolution microscope technique, which provides about 300 nanometer of lateral resolution. And nowadays, the SMLM is widely used to investigate biological structures and has revealed a lot of details, as we can see in these examples. And this single molecule localization microscopy requires photo switchable fluorophore on the target structure. So we use the SYN3B cells with an extra copy of target genes labeled with a fluorescence protein as well as the flag epitope. And a well-known photo switchable fluorophore, Alexa 647, is labeled on the flag tag by immunofluorescence labeling. And here I summarize the conditions for sample preparation and the SLML imaging. Briefly, I grew up the cells in SP4 knockout media for a few days based on the phenolate indicator. And harvesting of the cells was carried by centrifugation under these conditions. And for the immunofluorescence labeling, I fixed the cells with 4% PFA, and I permeabilized the membrane with tritone or 2020. And the IF labeling follows a standard protocol commonly used for the mammalian cells. And the single molecule localization microscopy is carried in the imaging buffer, contains glucose oxidase-based oxygen scavenger, as well as a proper amount of small sires to induce the photo switching. And these protocols sometimes gave a fair imaging result, but are not reliable yet. So as I need to further optimize these conditions, please let me know if you noticed any weird steps in this protocol. And our first target was the PTSG protein. And the PTSG is a membrane transporter of glucose. And a previous proteomics study estimate there are about 800 copies of PTSG proteins in a JCVI-syn3A cell. And in other organisms, the PTSG proteins are mainly located on the cellular membrane, as expected from its role. And to visualize the PTSG proteins, we used a JCVI-syn3B strain that expressing PTSG labeled by flag epitope as well as the M-apple fluorescence protein. And when the sample was well prepared, we could observe well-localized fluorescence signals between M-apple and Alexa 647 labeled on flag tag. And the single molecule localization images provided more structural details from the Alexa 647, and the result generally showed a high level of cell-to-cell variance. And here are some more examples for the PTSG distribution in JCVI-syn3B. And here the gray ones are the wide field images from MFO, and they are merged with the single molecule images represented as magenta. And it seems majority of PTSG proteins are located on the cell membrane, although there are a few exceptions. And they are clustered and distributed sparsely in these example images. And 3D SLML imaging provided a little more details. Here, the different colors means different Z positions of the protein. So the PTSG proteins do not evenly exist on the membrane, but they covered only a sub-region of the membrane. And when I quantified the number of localizations, the average number was about 1,300, which is way smaller than an expected value. So because given the copy number of PTSG from the proteomics study and the multiple secondary antibodies on a single PTSG protein, we may be able to expect at least 5 to 10 for larger localization numbers per cell. So this quantification result suggests a not optimized sample preparation condition, which might affect the sparse distribution of PTSG proteins. Another target is the FTSC protein, which are known to play a role in cell division. And at the time of division, the FTSC proteins forms a ring-like structure called Z-ring, as shown in this figure. And so we wanted to check the structure of FTSC in JCVI-syn3B cells to provide some pieces of evidence about the division of minimal cells. And from the 2D single molecule localization images, we could observe some partial ring-like structures, but not a fully composite ring structures. In JCVI-syn3B cells, the target FTSC proteins are expressed from an extra copy of the genes with a weaker promoter. So this may limit our observations for the Z-ring structure. And in these two examples for 3D single molecule localization imaging, the upper one is quite similar to the 2D result, but the lower one showed a cell filled with FDSC proteins. And so again, we saw the heterogeneous behavior of target proteins. And one possible reason might be a cell growth dependent regulation of those proteins. Since the size of the minimal cell tends to be proportional to its life cycle, maybe direct measurement of the cell membrane help to explain the heterogeneous protein distribution. And it also provides a reference structure that helps to interpret the distribution of proteins. So yeah, among the various membrane staining dyes, we tested the SPDIOC that has been confirmed to stain the membrane of minimal cells and has a comparable spectral property to our optical setup. And after checking this dye can efficiently stain the membrane in our hand, we later did explore the imaging conditions to induce the on and off transitions of the dye, and under the presence of small sires such as ME or beta ME, SPDIOC showed efficient photo switching behavior like this. So we tried to co-visualize the cellular membrane and FDSC proteins by using SLML imaging. And here, the cyanine ones are the membrane probes, and the magenta ones are the FDSC proteins. And again, the FDSC proteins are clustered and located in proximity to the membrane in both 2D or 3D measurements. And unexpectedly, the membrane showed highly disrupted structures with a low number of localizations that hindered the precise interpretation of FTSC protein distribution. Probably the permeabilization step for the immunofluorescence labeling can interfere the intact membrane organization, and the simulation work predicted half of the cell surface may be enriched with membrane proteins. So increased concentration and the increased concentration of membrane probe without the permeabilization step clearly increase the number of localization and allow to get the cell diameter. And the measured diameter has an average value of 800 nanometer for JCVI-syn3A, which is slightly larger than previously reported values. So in summary, we are trying to investigate the structural details of some proteins inside the JCVI-syn3A and B cells by using super resolution imaging. And both PTSG and FTSZ showed clustered and membrane close distributions with high cell-to-cell variance. And SPDIOC allowed direct visualization of the cellular membrane, but the permeabilization step severely disrupt the membrane organization. So to proceed further, we need to optimize the conditions for the sample preparation as well as for the imaging to get more reliable measurements. And one of the best way could be the introduction of photo switchable or photoactivatable fluorescence protein instead of numer 1 plus FLECTEC. It will allow direct single molecule localization imaging from those fluorescence proteins without the permeabilization step and even in the live cells. So yeah, this is it for today, and I'd like to thank all our collaborators for their great discussions, as well as all of you for your attentions. And now I'll take any questions.
Nice talk. You are showing me things about the cell that I'm hadn't been able to see before using non-electron microscopy. So this is a real advance. I think you and Take-Hip and also Joshua, who will talk next, this is a real improvement in our ability to image the cell. Any questions, anyone else?